biochemistry, biophysics, then later professor of biochemistry and basic medical sciences at Washington State University from 1972 to 2008. Since 2008, he's professor emeritus of biochemistry and basic medical sciences at Washington State University. And he's received 9 international honors for research in environmental medicine and authored 103 professional publications. His first paper on EMF was published in 2013, presenting the basic method of action, and was cited over 350 times in the literature. And there's widespread recognition of this work. So please give a warm Academy welcome to Dr. Pall.
My talk will focus on several different things. One is 9 extremely well-documented effects of non-thermal electromagnetic field exposures. The second is why these 9 have such dire implications, and I'll only talk about really 2 of those in some detail. There are, one of the things that's very important about EMFs is the electronically generated EMFs are coherent. They produce EMFs with a particular frequency, with a particular vector direction, with a particular polarity and phase. And for all of those reasons, they produce vastly higher electric and time-varying magnetic forces than do natural incoherent EMFs. So in comparing them, there's really no comparison. And it's the biological effects that are produced through these electric and time-varying magnetic forces, and both of them are important in producing biological effects. Coherence, by the way, is essential for using EMFs for wireless communication. So the reason that we're exposed to these so much is exactly the same reason why they have so many biological effects. The EMFs work via voltage-gated calcium channel activation, and I'll talk about how this occurs with the coherent EMFs being extremely powerful in producing that. And then there are multiple findings that show that the safety guidelines are fatally flawed. And I will talk about 5 different brain-related effects in addition to the one effect that I'll talk about with this first set. There are, in total, many different nightmare scenarios in this talk, so be forewarned. We have a large number of effects that have been widely documented in many different review articles in the scientific literature. So lowered fertility is one of them. There are effects on the structure of the testis, the ovaries. Those have been demonstrated in animal studies, in experimental animal studies. There's lowered sperm count, sperm quality, lowered sperm, lower numbers of egg follicles, increased spontaneous abortion, and lowered levels of each of the 3 sex hormones, lowered libido. So almost anything you can think of that impacts human reproduction is negatively impacted by this. The things that have been most studied have to do with male reproduction. And so there have been studies done, for instance, on human sperm, where you take a human semen sample, divide it in half, expose 1/2 to things like cell phone radiation or Wi-Fi radiation. And what happens is you get vastly lowered motility. You get a large degradation in the structure of the human sperm. These occur very rapidly. And there are other changes. And there are also attacks on the DNA of the sperm. So these are extraordinarily important. And I think that, you know, so one of my nightmare scenarios basically is that fertility will crash to 0 or pretty close to 0. And we're already seeing very large decreases in human reproduction. There are quite a number of countries in the world that have reproduction at or below 50% of replacement levels, which means you get a drop in half with each generation. And the fertility rates keep dropping lower. In an animal study, it was shown you can get a drop in reproduction to zero very quickly with EMFs of the sort that we get exposed to. So, you know, this is a real possibility, and it's one nightmare scenario. We may simply become extinct because of reproductive crashes. Oh, let's see, I'm sorry. Okay, second one has to do with these neurological, neuropsychiatric effects. There are 35 reviews on these. The part of it that I'm concentrating on, these various neuropsychiatric effects, are actually only in about a third of these reviews. But, you know, you get, and we've had, you know, there's a lot of discussion about insomnia. Insomnia is the #1 effect of EMFs in humans. This is very well documented. I can't sleep, I'm tired all the time. We know it's caused by EMFs of the sort we get exposed to all the time. You get depression, you get headaches, you get lack of concentration, anxiety, stress, memory dysfunction. And what do? We put Wi-Fi in schools, and we expect people to be able to learn when they have memory dysfunction and cognitive dysfunction. Does that make any sense? Doesn't make any sense to me. You get cellular DNA damage. So I just want to say, with regard to these things, these neurological, neuropsychiatric effects, there is good evidence from occupational exposure studies that were done way back in the 1970s and 1980s, when we had very few exposures of any sort, and where it was shown that these things develop cumulatively. That is, the longer the times of exposure to a particular type and intensity of exposure, the worse things got. So the question and the fear here is that our brain function is progressively deteriorating over much of the world, and this may then help explain many of the things that I think all of us see every day. Cellular DNA damage of 3 different types has been extensively documented. That can lead both to a degradation in the gene pool, in the structure of our DNA, that is essential for any propagation of the species, as well as in terms of cancer causation. We get large increases in apoptosis, that's programmed cell death, and that's important for these reasons. And I'm sorry to the people over here that I'm just, can you see this all right? Okay, good, thank you. You get oxidative stress and free radical damage, which has roles, as I'm sure you know, in essentially all chronic disease. We get endocrine, that is hormonal effects. And as far as I can tell, essentially every single hormone system in the body is impacted. Some of these directly and others indirectly. You get excessive intracellular calcium, which I will argue is the underlying cause of almost everything else. You get cancer. There are 44 reviews on cancer, which basically implicate EMFs in causing everything, each of the many events that are involved in carcinogenesis, including tumor promotion, progression, tissue invasion, and metastasis. The only reason why we're still discussing this stuff is because the industry has so much propaganda power that they argue that there's not much evidence. In fact, there's vast amounts of evidence here. There are cardiac effects on the electrical control of the heart, where you get changes in, you can get tachycardia, rapid heartbeat over time. You can get bradycardia, slow heartbeat. You get arrhythmias in both situations, and arrhythmias can cause sudden cardiac death. There are animal studies that have shown that EMFs can cause sudden cardiac death. This is another thing that's epidemic in human populations now that was very rare in previous times. We have, in summary, in those, there are 219 bodies of evidence. These are bodies of evidence in those different reviews. And that doesn't, you know, there are many of these reviews have more than one body of evidence indicating that we have a serious health-related effect produced by EMF intensities far below the current safety guidelines. So anybody who wants to argue for the safety guidelines must show that these 219 bodies of evidence are deeply flawed. Okay, now, I guess I mentioned this before. We do have evidence now for cumulative effects on the structure of the brain, of the testis, of the heart, and possibly a number of other organs. The testis changes were shown to be caused by oxidative stress. So the idea is you can get cumulative changes in structure, what's sometimes called tissue remodeling, which then produces long-term dysfunction. The DNA changes are inherently cumulative and irreversible because they involve mutations. So that's an issue as well. We do get cumulative effects, and they're very important. So, you know, the idea that most people have is, well, you stop exposure and everything goes back to normal is simply untrue. Okay, so how do we get non-thermal effects? That is, effects that do not involve heating. And let me just say, the industry and the regulatory authorities claim that everything is, the only thing that these things can produce is heating, that everything is a thermal effect. But the non-thermal effects have been known going back 50 years and more in the literature. And what I've shown is that there are large numbers of studies, and I've cited 30 of them in my papers, where microwave or other frequency EMFs, the effects of them can be blocked or greatly lowered by using calcium channel blockers. These are channel blockers which are specific for blocking voltage-gated calcium channels, or VGCCs. Okay, and so what that tells you is that most, if not all, of these effects are produced via activation of these voltage-gated calcium channels, these VGCCs. And so these effects occur at many different frequencies: millimeter wave frequencies, microwave radio frequency frequencies, intermediate frequencies, extremely low frequencies such as 50 or 60 hertz from our power wiring, and also even static electrical fields and magnetic fields. They can all act via VGCC activation to produce biological effects. And that's a very important and surprising finding, that there would be a single primary target of these diverse EMFs. There are other voltage-gated ion channels, sodium, potassium, chloride channels, TPC channels, which are important in plants, that are also activated. Each of these contain a similar structure called the voltage sensor, which I will argue is the direct target of the EMFs. Okay, so this is a structure in these channels that is where these forces that I talked about before, the electric forces and the time-varying magnetic forces, can have direct impact on the voltage sensor, and thus open the channel and cause things to be activated, cause those channels to be activated. Now, these other channels are also activated. They have relatively minor roles in producing effects. So most of the effects go through the calcium channels, the VGCCs, but you do get at least some effects from these. And let me just say, the sodium channels may actually be quite important in terms of producing pain responses. That may be one of the things where a different channel might actually be involved in producing important effects. Now, so electronically generated EMFs are coherent, and as I said before, being put out with a particular vector direction, frequency, phase, and polarity. And for these reasons, these produce vastly stronger electric and time-varying magnetic forces than do most natural EMFs that are incoherent. And that was published in this paper here early this year. The millimeter wave electric field is rapidly absorbed in body tissues, but the magnetic fields are very highly penetrating. So the industry claims that millimeter waves cannot produce penetrating effects, but they do. There's empirical evidence showing they do. And so I argued in this paper that those are produced by the time-varying magnetic forces, which are very highly penetrating. So the idea that the industry pushes is that 5G using millimeter wave frequencies can't do anything except on the very outer parts of the body is simply untrue. And so you do get much more highly penetrating effects of both millimeter waves and microwaves acting through the magnetic fields. I'm using this. This is a model of parts of these voltage-gated ion channels. This particular one comes from the potassium channel. Doesn't really make any difference on that. What these little squiggles are alpha helices in the proteins, and so each of these channels have 4 different domains which are repeated. This one, this one, this one, and that one are essentially identical, almost identical to each other. And each of those 4 domains has a particular helix called the S4 helix. That's the one that's got these lines drawn to it, okay? And so here's an S4 helix, here's another one, and here's another one. And what these things do when the channel is triggered to open is they ratchet out away from the center. So they move in this direction, that direction, and this direction. So they move out away from the center, which is right here. And when they do that, they pull on 2 other helices, S5 and S6 helices, and then you get a conformational change. And the thing pops open, and you got a channel open. Okay? So it's the forces on these things, and these each have 5 different positive charges on them. And it's the forces on those 5 charges on each of these 4 helices that is responsible for opening the channel. And so what I argue is that it's the EMFs that are approximately perpendicular to the plasma membrane where these things are sitting that produce similar forces on each of those S4 helices at the same time, cause them each to ratchet out at the same time, and thus open the channel. And that's the basic mechanism which I think is going on here. And both the electrical forces and the time-varying magnetic forces can put forces on those electric charges. So, so I think both of those are involved directly activating these channels. What's interesting and important is that this structure itself just opens a channel. It doesn't determine what ions go through it. There's another structure in here called a selectivity filter that's down below this, and it then determines which ions can go through the channel. And we know that because, for instance, if you take the amino acid residues that are involved in the calcium channels and mutate them to different amino acids, you can convert a calcium channel into a sodium channel just by doing that. And that was done back about, over 20 years ago now. So we know a lot about how this whole thing works. And now, so basically, we have a lot of information about how it works, and now the question is, well, okay, how do you get biological effects? And the first thing that happens when you activate these channels, these VGCCs, is you get large increases of intracellular calcium. And so you get huge amounts of calcium flowing into the cell for two reasons. One is that calcium levels outside the cell are something like 10,000 times higher than they are inside the cell. But there's also the electrical gradient in the plasma membrane that also drives calcium in there. So there's something like a million-fold electrochemical gradient driving calcium into the cell. So you open these things, you get huge amounts of calcium ions flowing into the cell. So Ca2 plus I is intracellular calcium. Right? And it then has a lot of effects. Now, how do you get, and so what I'm going to talk about now is how you get pathophysiological effects, the damaging effects. It mainly goes through two pathways. You can get excessive calcium signaling. Calcium signaling is very important in cells. And when you get way too much of it, that can produce pathophysiological effects. And in fact, and you also get increases in both nitric oxide and superoxide. These two react with each other in a what's called a diffusion-limited reaction. So anytime one of them bounces into another, it reacts extremely rapidly to form peroxynitrite, which is this guy. Peroxynitrite is not a free radical, but it breaks down to form highly reactive free radicals and produce oxidative stress. And these all act to activate NF-kappa B, which produces inflammation. So you get a lot of oxidative damage and inflammatory damage, and those things have very important roles in producing pathophysiological effects from EMFs. And they also, by the way, act to produce mitochondrial dysfunction, which also has roles there as well. Now, there is, in addition to that, there is a therapeutic pathway here, which is thought to be activated by modest increases in intracellular calcium, and that works through nitric oxide. Okay, so there's been a lot of discussion of nitric oxide at this meeting, and nitric oxide in turn acts through the soluble guanylate cyclase and cyclic GMP, and to activate a protein kinase, sometimes called protein kinase G. And that, in turn, activates something called NRF2. NRF2 is a transcription factor which controls something like 500 genes in the genome. And it's NRF2 that primarily produces these therapeutic effects. So this is the sequence that we think is going on in producing therapeutic effects. Now, one of the things that's very, very important here is the following: that these therapeutic effects have one kind of one kind of thing, and they produce antioxidant effects, they produce anti-inflammatory effects. They basically produce effects that are opposite the ones that are produced by the peroxynitrite pathway. And those two pathways not only produce opposite effects, but each of these pathways, each of these two pathways, the therapeutic pathway and the peroxynitrite pathway, inhibits the other via multiple known mechanisms. So what you have here is kind of a switch between one and the other, and you can get extraordinary differences in responses depending on which of these is predominant and which one is not. So let me just say, the industry claims, well, we only pay attention to things that are consistent. Well, in fact, things are consistent, but when you look at some kinds of EMFs, you get one effect, and in other kinds of EMFs, intensities or types, you get the other effect. And they're almost exactly opposite each other. And the industry ignores all of these data just because it's convenient to do so. let me say a couple other things about this. There's been a lot of discussion about nitric oxide being a favorable response, and it has this favorable response. But nitric oxide is not a unmixed blessing. It has negative effects in addition, and some of those go through the binding to cytochromes. So cytochromes are important in mitochondrial energy metabolism. They're also important in biosynthesis, and they're also important in detoxification of toxic compounds. So nitric oxide inhibits all of those things by binding to these cytochromes. And so one of the things that's been discussed in this meeting is vitamin D. Vitamin D has to be converted into, 25-hydroxy vitamin D in order to be active. The enzyme that converts vitamin D into 25-hydroxy vitamin D is a cytochrome P450. So it's inhibited by nitric oxide. So you can see how these things become much more complex than a lot of people have proposed. And now the other thing nitric oxide does, as I said before, is it acts as a precursor for peroxynitrite. So it then activates this pathway as well, and so that has major impacts as well. So nitric oxide is not a unmixed blessing. I mean, it has problems that are associated with it as well, and one has to be very careful about that. Let me just say one other thing about this. There are many, many health-promoting factors that raise NRF2. Nitric oxide signaling is one of them. There are other ways of getting the same favorable response. Okay, so let's go on here. And second thing that's very important is that modulated EMFs, these modulate an underlying frequency, and so they produce large pulsations in the magnitude of the EMFs at each terasecond of time, if you will, and they produce in most cases much more biologically they're much more biologically active than our continuous, that is non-pulsed EMFs of the same average intensity. So average intensity tells you nothing about biological effects, but average intensities are what are used in the safety guidelines to predict biological effects and to tell us supposedly when we're safe and when we're not. So that's another piece of complete nonsense that occurs here. So wireless communication devices, with the exception of FM radio, communicate via modulation. And the smarter they are, the more information they carry, the more they pulse, and therefore the more biologically active they are. And so so we are in a continual process where so-called smarter and smarter devices are being put out that pulse much more and therefore, at least potentially, and I believe clearly actually, are much more biologically active. And this is completely ignored by the industry. And so this is a very, very important issue. 5G is designed to be extraordinarily highly pulsed in order to carry vast amounts of information. But even the preceding, things that have come out before 5G, like 4G and smart meters, which also do a lot of pulsation, may be quite dangerous as well. And even things like 3G and 2G show biological effects, including some cases major biological effects. So we're in deep trouble. This is a table of the 1998 ICNIRP safety guidelines. ICNIRP is the International Commission on Non-Ionizing Radiation Protection. It's an organization that was basically set up by the industry to predict what our safety guidelines should be. The safety guidelines are all average intensities averaged over either 6 minutes, as these are, or in some cases 30 minutes. So we're always looking at average intensities over a long time period. And in addition to that, the cutoffs, the allowable levels, are based on heating. They're based on how much heating you get. And so it ignores all the non-thermal effects that we know about. There's some other peculiar things about this, and that is the whole body levels are much lower than partial body ones. So here you have about a, about a 50-fold difference in allowable levels for certain parts of the body versus whole body. But the cells of our body, this is a cellular response, don't care whether the whole body is irradiated or whether just part of the body is irradiated. You get the same response in the cells. So these things make no sense. There are also occupational exposure studies that are about allow five-fold higher exposures. But many of the people in these occupations don't have any better way of protecting themselves than we do. So, so there are all kinds of peculiarities here. And let me just tell you one other thing about this. They average these exposures, making the false assumption that EMFs are scalars rather than vectors. And the way in which you average a vector is totally different. The mathematics is totally different than the mathematics of how you average scalars. So even their averages are incorrect. The physics is incorrect, and therefore the mathematics is incorrect. So this whole thing is, no, I won't even try to tell you what it is. Now, okay, so now I just want to give you some information here. How are we doing for time now? Right? Yeah, okay. Okay, so I need to go a little bit faster than I've been going. There are a number of studies that have shown that what are called nanosecond pulses. These are just pure pulses that go up and come down in a period between 1 nanosecond and 1 microsecond. So this is about a 1,000-fold range. But they're all really quick. They go up and come down. And they produce effects over and over again. And in some cases, it's been shown that you can block those effects with these calcium channel blockers. So we know they work via VGCC activation. If you take a typical, say, 40 nanosecond pulse, and you average it over 6 minutes, which is about 10 billion times longer, then, of course, what the industry says is that the average intensity is decreased by a factor 10 billion, and they say, well, it's too low to do anything. But in fact, it does things all the time, over and over and over and over again. So, so this is all nonsense. And I think I'm going to skip this other part, but it's interesting, but I don't think I have time to talk about it. There are also effects. There have been studies of pairs of nanosecond pulses, and these are quite interesting because what they show is if you have 1 nanosecond pulse and then you have a second pulse within a few microseconds of the first one, the effects of the second pulse depends on the polarity of the second pulse. So if the second pulse has the same polarity as the first one, you get super additive effects. You get much more effects than the sum of the two individual effects. If, on the other hand, the second one has the opposite polarity, you get partial cancellation. You get much lower effects than you get from the first pulse alone. So this tells you a couple of things. One is that the polarity, which is one part of the coherence that we talked about before, is extremely important, right? and that the, you know, if the second pulse has the opposite polarity of the first, it actually gives you a lower effect than the first one alone. And so you can see the complications of these things. These are extraordinarily complicated. It is almost impossible to make predictions about them. And this tells you something about the coherence effects, because this is one of the coherence effects. Okay, I'm just going to go through these very quickly. There are three other fatal flaws to the safety guidelines. One is the effects you get is a function of what cell type you're studying. So the safety guidelines simply based on physics. They just ignore all the biological heterogeneity. But we know you can't do that because different cell types, in fact, respond differently. There are intensity windows, that is specific ranges of intensity that produce maximum effects. But when you go either lower or higher, to lower or higher intensities out of that window, the effects drop way down. So there is no, you know, there's no obvious, understandable dose-response curve here, okay? You know, you can't say that because you've got more exposure that you always get more effects. Often you do, but you don't always, and there are many, many times when you don't. Okay, there are also frequency windows, specific frequencies that give you extraordinarily high effects at very, very low intensities. And these have been ascribed to resonance interactions with specific targets. So all these things say basically this is an extraordinarily complex system. And I don't think there's anyone in the world that can make any kind of decent predictions about what you're going to get from these things. I mean, so all these things say the safety guidelines are bogus. And so we have a multi-trillion-dollar set of companies, the telecommunications industry, all of whose claims of safety are based on failed safety guidelines and therefore simply fraudulent. Okay, now what I'm going to do now is to go back to brain-related effects. Okay, so we talked about the neuropsychiatric effects and why I was concerned about those and why that's one of the nightmare scenarios that we have to worry about with regard to these things. Now I'm going to talk about 5 other effects and see how we're doing here. And actually, I guess we're doing all right. One of them has to do with EEGs, the electroencephalogram. There are 38, so these are experimental studies on human brain EEGs where it's been shown that, that low-intensity EMFs of different sorts, including cell phone radiation, including Wi-Fi radiation, but including a lot of other things, produced EEG changes in the human brain. So this is a measure of electrical activity in the brain. And so those studies, in and of themselves, you say, well, maybe we don't have to worry that much about it. But the other things that I'm going to talk about are all things we do have to worry about. And one of them has to do with Alzheimer's and other dementias. Another one having to do with autism and also ADHD, that I will talk about. And now here's an additional one, which has to do with addiction. The changes in the human brain, well, actually, these have been studied in animal brains. The changes in animal brains that occur following EMF exposures are extraordinarily similar to the changes that are produced by addictive drugs. And so I'm going to argue, first of all, that EMFs are physiologically addictive, and that in turn causes us to become addicted to our devices that put out EMFs. Okay, so that's another nightmare. So these, yeah, I think you can see this one, that one, and this one. They're all nightmare scenarios. then the last one is electromagnetic hypersensitivity. And I hope I'll have time to talk about that. Okay, so let's go through okay, so we talked before about these neurological, neuropsychiatric effects, and that these produce cumulative effects. And if these lead then to a major degradation in our brain function, which I think has already occurred, the integrity of our whole social fabric will begin to disintegrate, and we'll just go into chaos. And the EEG studies, and I think maybe I've said enough about these. Is that right? Yeah. These are produced by millimeter waves. They're produced by microwaves, and so a number of different frequencies, and quite a number of things that we are commonly exposed to produce changes in the EEGs in the human brain. Okay, let's talk about Alzheimer's disease. There is a hypothesis of Alzheimer's disease. Alzheimer's disease, I'm going to abbreviate AD. That's called the calcium hypothesis of AD, which argues that excessive intracellular calcium is the central cause of AD. Okay, and as I showed you before, the first thing that happens when you expose the brain to EMFs is you activate the VGCCs and you get increases in intracellular calcium. So it therefore is highly plausible that, that EMFs can cause AD. Now, it turns out that there are, in addition to these two considerations, 16 additional types of evidence which argues, in fact, that EMFs are causing neurodegeneration, and specifically including Alzheimer's and including a very early onset Alzheimer's. And so that's the, and you know, these are, these are complex considerations, and I'm not going to talk about all the evidence here because I don't have time. But basically, so we know that, for instance, you get neurofibrillary tangles in Alzheimer's disease, and you get those in by EMF exposures in animal brains. The proposed cause of these is hyperphosphorylated tau protein, which is produced by calcium signaling through these mechanisms. So, so we have highly plausible mechanisms by which excessive intracellular calcium can cause these neurofibrillary tangles and hyperphosphorylated tau protein. We have epidemiological studies. I believe there, I can't remember, something like 15 of those, that report that people who are exposed to EMFs occupationally have higher incidences of Alzheimer's disease. We have very young people exposed to things like Wi-Fi and cell phone radiation over hours a day, who develop what have been called digital dementia. So these are very much dementia-like effects. We don't know whether these have similar physiology to AD or not. It's never been looked at, okay? So these things do occur. They occur in very young people. And then the question is, what's the mechanism, and does it relate to what's going on with Alzheimer's disease? Now, EMFs produce large increases in the amyloid beta protein. This is thought to be the specific protein that's involved in causing Alzheimer's disease when it is elevated in the hippocampus and some other parts of the brain. And so you get increases in A beta in isolated neurons, and you also get increases in A beta in animals in their brains following EMF exposures. Low-intensity EMF exposures not anything unusual. And it's thought that oxidative stress has a key role in Alzheimer's disease causation. Anyway, so these occur in animal brains. And now I'm going to talk about a few specific animal studies, which I think are brilliant studies and are really things that are worth looking at. If you get a chance to look at these papers, you should do that. There was an interesting paper published by El-Swefi et al. In 2008. I just discovered it very recently, just a few months ago, when I was doing some searches. What they showed was that rats who were exposed 2 hours a day to very low intensity mobile phone base station radiation. This is what we call this cell phone tower radiation. It's a different name for the same thing. Most of the world calls it mobile phone base station radiation. 2 hours a day, low intensity, developed over 4 weeks, which is about 2.7% of the lifespan of the rat. Massive neurodegeneration in the brain, with 34% of the cells of the brain dying in that 4-week period. They measured 11 changes that were occurring that contributed to this neurodegeneration. And each of those 11 were greatly lowered by amlodipine. Amlodipine is one of the VGCC calcium channel blockers. So what does that say? It says that all 11 of these changes are produced via activation of the VGCCs by the EMFs. And there were also 4 observed changes that were also greatly lowered by this calcium channel blocker. So in total, you got 15 different things, and all of them are produced largely or entirely via VGCC activation, produced by this mobile phone base station radiation. And this is, as I said, this all occurs in roughly 2.7% of the lifespan of the rat. I mean, this is absolutely stunning. So you can see that, and let me just say, and I'm not going to show you data on that, all 11 of these things, these measured changes, can be explained through the mechanisms I talked about before in that figure about how you get effects through calcium signaling and through the peroxynitrite pathway to produce pathophysiological effects. All 11 of these things can be produced through those pathways. Okay, now let's talk about another couple of studies, also done in rats by a different. By the way, El-Swefi, that's a group in Egypt, which is not a country that one thinks of as producing world-class science, but that thing is world-class science, I have to say. There are 2 studies that came out of China, and China is producing some very good science nowadays, regularly. What they used were these were basically nanosecond pulses, 10 minutes. Okay, and they produced nanosecond pulses in young rats. And what they found was if they gave those pulses, 100 pulses, 1,000 pulses, 10,000 pulses, 10 milliseconds apart from each other, at 2 months of age in a rat, which is sort of adolescent rats, because they develop rapidly. And I'm sorry, I don't think, oh, okay. This is the one, I'm sorry, did I miss something? Maybe I did. No? I'm sorry, I left something out of your slides. When they did that, they got AD-like effects 18 months later. So there's a 20-month-old rats. Rats live 36 months. These are still relatively young rats. This is like 41, 42-year-old humans coming down with universal Alzheimer's based on EMF pulses that occurred once to the animal, just once, a series of pulses. Now, if instead of giving the pulses once, you give them once a day starting at 2 months, then you get 100% AD-like effects at 8 months. 8 months later, rather. So 10-month-old rats. These are like 21-year-old rats coming down with universal, very, very, very early onset AD. And these rats have high levels of A beta in their hippocampus. They have high levels of the amyloid precursor protein, which gets converted into A beta in the hippocampus, and they have high levels of the protease, base 1, which converts APP into A beta. So they have all three of the characteristic defining mechanisms involved in Alzheimer's disease in 10-month-old rats, equivalent of roughly 21-year-old humans. 100%. Almost unimaginable. These are incredible nightmare scenarios. And now I want to talk about autism. And I better be quick. How much time do I have? Not much. I guess about 7 minutes is what I've got. Is that right? Huh? Yeah. Okay. So what I'm going to argue is the following, that autism can be caused both by EMFs and by various chemicals, and that both the EMFs and the chemicals act via excessive intracellular calcium. And what I'm going to do is to argue that the way they act is they act to disrupt the formation of synapses in the developing brain. And they do that via each of these 5 mechanisms. There's regulation of dendritic outgrowth, of synaptic formation, synaptic maturation, synaptic elimination, and then there's something else called MACP2 function. All of these have important roles in controlling the formation of synapses in the developing brain. And each of these 5 mechanisms is controlled by intracellular calcium. So when you have an artificial rise in intracellular calcium in the developing brain, you will disrupt the regular formation of synapses. And we know that, that in autism, that one of the things that happens is the connectivity of different parts of the brain changes compared with normals. The way in which one part of the brain is connected to another part is changed. And of course, what changes those things? The synapses that connect these neurons to each other. So we have that, but we know that from genetic studies that excessive VGCC activity can cause autism. There are various genetic studies that have shown that, but there are also effects of chemicals. And I'm going to argue that the chemicals act through the NMDA receptors, through increases in the NMDA receptor activity, which in turn produces increased intracellular calcium, and then also can produce similar effects. And there are other things that are going on here that I don't have time to talk about. But I want to say, so this is some of the genetic evidence that I mentioned before. And I want to talk about the chemicals because I think this ties up with a lot of the interests in this group, okay? So there are 7 classes of chemicals which I argued in a 2009 review that I wrote on multiple chemical sensitivity, can all act to produce increases in NMDA receptor activity, and therefore produce increases in intracellular calcium. And they act along known pathways. And I'm just going to, I'm going to talk about one of these, the mercury one, which actually acts via two sets of mechanisms. I'll just talk about this one. And so mercury can act through this pathway. When you have a double negative, which is what these two are, what does that give you? It gives you a positive. So all these pathways lead to increased NMDA activity, okay? All of them. Some of them have double negatives, some of them are just positives, but they all do that. And so you can say, well, okay, in theory, this can act in that way, but how do we know that that's important? And the answer is that people have done studies on the toxicity of these chemicals in animals, and they have shown that the toxicity is greatly lowered by using an NMDA antagonist, by using something that greatly lowers the NMDA activity. So what do we know? We know all these chemicals act, in fact, in animals to produce excessive activity here and therefore increases in intracellular calcium. And that's been shown, by the way, with methylmercury, okay, which is the more active of these, are much more active than mercury metal, as I'm sure you people know better than I do. So anyway, I think that there is reason to think that both chemicals and EMFs are working to produce autism through this mechanism. What I'm also going to, my opinion is that the EMFs are more important than the chemicals. And the reason that's my opinion is that most of those chemicals that I showed in that other table, or in that other figure, there were huge increases in production following World War II, and therefore huge increases in exposures following World War II, but in that 30-year period. But the autism epidemic really didn't get started until right after that, and when we started getting more and more EMF exposures. So my opinion is that EMFs are probably the main driver of this, but I think this is something reasonable people may differ on, and I'm not saying that's a hard decision. But in any case, these mechanisms mean we should be working actively to reduce exposures to both of these. And right now, the EMFs, we have increasing exposures at incredible rates. I mean, it's just stunning. Okay, so, all right. Addiction. I think I won't have time to talk about EHS. There are a whole series of mechanisms that are essential for cocaine, opiate, nicotine, and amphetamine drug addiction that are also produced by EMF exposures. And so I've listed the most critically important of these, okay? So in drug addiction, there are two VGCCs, CAV 1.2 and 1.3. These are the two most important, what are called L-type VGCCs in the brain. And they are known to each have distinct roles in causing drug addiction. They act sequentially in the process of drug addiction. So one of them acts first and the other one second. And the combination of the two then produces drug addiction. One of the things that happens as a consequence of that mechanism is you get increases in release. And so I should simply point out, we know both of these are activated by the EMFs, right? So that's so the EMFs can do this, and therefore can, at least in principle, lead to addiction to that EMFs can be addictive. In addition, increases in the endogenous opioid dynorphin occurs, Much of the neurological changes that occurs in EMF following EMF exposures, these were done in rodents, were shown by Henry Lai from the University of Washington here to be produced by dynorphin stimulation of the opioid receptors in the brain, because you could block the effects with an opioid receptor antagonist. So clearly, this effect is a very important one. And so what this shows, these studies that were done by Henry Lai, these were published over 25 years ago. What was shown was that you could block much of the neurological effects by using an opioid receptor antagonist. That tells you that you not only get large increases in dynorphin, but that's very important in terms of the whole brain function changes that you see from EMFs. The second thing which he and his colleagues showed was that EMF exposure, the effects of these EMF exposures, the dynorphin release and so forth, could be learned by classical conditioning. In other words, when you pair the EMF with something else, like a bell in Pavlov's dogs or some other thing, the animals will learn to produce that effect, that response to the paired signal, whatever it is. And so what this does then is it provides a mechanism by which we can become addicted to devices that produce EMFs. Physiologically addicted to the devices, to our smartphones, addiction to these devices is well accepted now in the addiction literature. Addiction to the internet is well described in the addiction literature. And of course, most people connect up to the internet using Wi-Fi. Those are all things truly extraordinary. Dr. Fry, who was also working roughly at the same time that Dr. Lai was working, showed that there are increases in dopamine release, which has essential roles in the reward process in addiction. So all these critical things occur in addiction and in drug addiction, and they are also produced by EMF exposures and can be learned by classical conditioning to be produced by things that are paired with it. Now, so this is another nightmare scenario that almost every man, woman, and child on Earth are being exposed to. To a at least potentially addictive stress, all over the world, almost everybody. Now, I'm just going to raise 3 questions, then I'm going to quit. One is, and I'm not going to answer these questions. One is, did the telecom industry design devices to be addictive? After all, Henry Lai's studies were published over 25 years ago, and they certainly must have known about those. The second thing is internet addiction to different sites the basis of many of our problems? For instance, addiction to porn sites. People who use the internet, the Wi-Fi to connect to porn sites may well become addicted to those porn sites. Similarly, people who like to look at particular kinds of political sites of whatever frame might become addicted to those political sites. And so, the potential here is mind-boggling. I think the other thing I'm just going to raise, and that is that in the opioid crisis, that the connection between opioid drug use and EMF environments may lead to the EMFs actually leading to much of the opioid crisis in the following sense, that when people are in a high EMF environment and they're taking an opiate drug, what are they going to do? They're going to stop taking the drug, right? Because they already have plenty of stimulation. If they go back into now, if they go into a low EMF environment, they're probably going to start taking a lot of drug, right? Because they're going to have major withdrawal symptoms. And in principle, then, if they go back into a high EMF environment, they might have an overdose and die. Now, this is obviously an oversimplification for a complex thing. I'm not trying to say that that's all that's going on, but there may well be a major causal element of EMFs in causing the opioid crisis. And this is something that nobody's considering. You know, I mean, we've had, over the last year and a half, attempts to greatly decrease opioid drug use, you know, legal opioid drug use, and what's happened is the number of deaths from the opioid crisis last year went way up. It didn't go down. Anyway, okay, thank you very much.